Thank you both. Floor is open for questions from the committee. Let me, oh, I see Wiley going up. Go ahead, Wiley.
Oh, I think you've really already alluded to it, but I just want to be very explicit about it. Could you reflect on what you think is the optimal policy or guideline vis-a-vis CLIA in returning results?
Yeah, so I think as we go forward, any test where you anticipate returning the result, you should use a CLIA lab, and you should build that cost into the study. But there are many kinds of research that even I do. I have, 30-year-old samples in my freezer, and they will never be CLIA. And so I do believe that if you find something that is medically actionable and high penetrance and meets that standard, that you should offer the possibility of giving that non-CLIA result back and letting the clinical care then take over the clinical testing. Mercy Lorino just published a paper from a colon cancer consortium that I was peripherally involved with, where they returned, I think, 26, and they followed up in 12 months, and only 4 people actually did get a CLIA test, which is, I think, what Wendy said. Not everyone will get a CLIA test, but in order to use that, those people all had colon cancer, so maybe it wasn't a big surprise to them. In order to use the result in medical care, it really should be a CLIA test, and any doctor who's going to make a decision based on that test should make sure that they're not operating on a research test but a CLIA test. But again, as was pointed out in, I think, the last session, that we have actually never had a discordant result between the research and the clinical tests. The labs that we're working with, even for genomics, even the non-CLIA ones, are, like robots do almost everything. They have very, very high quality controls, but research labs will be very variable, so you always need that extra legally mandated step.
We're saying CLIA confirmation, or doing a lab and doing a result in a CLIA lab when you anticipate return is the optimal. But I think what you're saying also is that there will be times, one can foresee times, when one has a non-CLIA confirmed result that really should go back because of the nature of the information. The fact that people will then not necessarily go ahead and follow your recommendation to get a CLIA confirmation or go and do the appropriate, clinical confirmation, the fact that we have empiric data about that, does that change? Do we still say the results should go back?
So again, I think once you give the data back to people, they get to make their choice about whether they want it to be CLIA validated or not. In order for their physicians to use the information, it must be CLIA validated, and that's really on the physician to take that step, but it may not be useful to the participant to actually have it CLIA validated. Or we had cases among the people who didn't get it CLIA validated where they had relatives who did have the test. It was positive in a CLIA lab, and so pretty much they knew their result was correct. So it's not always necessary, really, for these participants to have them CLIA validated. But I think that it doesn't dissuade me in any way that they may not. I think it's up to them.
And it sounds like what we might take from this is also that the disclaimer needs to be clear. In other words, there needs to be a clear, the information going back to the participant needs to clearly explain the significance of it not being CLIA validated. Yeah, I'm so glad you brought that up, Wiley, because it is really important that when they get the non-CLIA result back, it's clearly labeled. This is not, this is a research result. It is not of clinical standards, and in order to be used in clinical care, we actually outright say we recommend that you have this redone in a CLIA lab.
I would add to that, though, I think there's probably some other things we need to be thinking about. There is nothing magical about CLIA laboratories. What's really important is having documented, reproducible processes in place in the laboratory. And I'm especially concerned about the cost-benefit analysis in this case. So Wendy's number that CLIA is about 3 times what a non-CLIA laboratory might be is a good example. We can get our same DNA extracted. with good chain of custody because it's completely automated at our place in a non-CLIA laboratory for about a third of the cost. And to think about the fact that we could do 3 times the number of samples is really a compelling point to me. The second point is, so that's the chain of custody issue. The second point is the actual validity of the test issue. And the examples that I cited where a CLIA test was just not as good as 500-fold deep sequencing of a gene, it's a shame to think about the amount of money that might need to be spent to repeat something where we've sequenced a gene 500-fold and know what the variants in that gene are, just because we need to meet some CLIA standard. So I think with those nuances, I think for sure we need to pay attention to the quality of the data, the chain of custody, and the quality of the assay, but nothing magical about CLIA.
Jeff.
Yeah, I guess a quick comment. I at least am going to need further clarification. My understanding was that the CMS folks in CLIA really prohibit any return of information about a test result, and that simply telling somebody that you've got an abnormality that needs to be confirmed in a CLIA lab is in itself prohibited by. So
it is also my understanding that CMS has made that interpretation. It does not, however, seem to stand up to legal scrutiny, and Barbara Evans wrote an 87-page legal treatise on this subject, and I wish she was here to tell you more about it. Her primary argument was a First Amendment argument that you cannot be, regulated out of communicating information, and fundamentally, this has never gone to court, right? I'm not a lawyer, but my understanding is ultimately these things get decided in court, whether they're legal or not. But I believe that Barbara Evans' case, I read it in detail, is a very strong case, and my institution allows return of non-CLIA results, and I believe it's institution by institution has looked at this. I know of institutions that don't allow that.
Okay, that's helpful. I do think we'll explore that a little bit more later. So I actually had a different question, which is, as a former primary care provider, I'm a little disheartened by the challenges of incorporating primary care providers as the disclosures of information. And it seems to me if research projects themselves have to set up the entire mechanism for results disclosure, that's a huge barrier. So I guess I'd be interested in any further thoughts you have on how to facilitate the use of primary care providers or engagement of primary care providers as team members in these circumstances to help disclose research results. Have you learned anything that might help us pursue that opportunity?
Yeah, we have. I think it's a process. So in the Emerge 2, when we did the pharmacogenomics return of results, there was a sense of being overwhelmed by the number of results that they were going to have to return because a very large number of people actually have pharmacogenomic variants that would make sense, but only in the context of them being exposed to the drug that they have a variant that might affect metabolism of. In Emerge 3, we found a very different approach, even though we've taken a primary approach of the genetic counselors returning the results. The primary care physicians now seem much more comfortable with the genes that are on, for example, the 109 gene panel for Emerge-seq. And I think it had as much to do with the volume of people that they were seeing. In Emerge 3, what we're seeing is on the order of 2.5% to 3% or so of our participants have a variant that actually needs to get reported, as opposed to in the PGX where it was a very much larger number. So I think part of it had to do with just the feeling of the volume of work that they needed to do and the lack of education that they had. As they've become more and more familiar with that, those barriers seem to be going away. And I think it's going to be all about provider education, providing more electronic tools that provide not only clinical decision support that enables the whoever is providing the results, but the ability to look at ancillary information. And so, for example, one of the things Emerge is doing, again, together with some folks from CSER as well, is looking at the use of info button technology in electronic health records to provide additional ancillary support for any provider that's providing information.
So I'd like to throw in, I'm not as optimistic. And this is not just genetics. I have lipid studies, and we were giving people back their lipid panels in a healthcare setting, and the providers mutinied against the patients getting their Lp(a) results back, which is an independent predictor of heart disease, but not one that's measured clinically that often. And they said they didn't, and this wasn't even they had to explain it, just their patients were going to get it, but they didn't want the patients coming in and asking them about it. Because why? They don't have time. They are generating RVUs, this is how they're paid, and they don't want added work that has nothing to do with their care of the patient, and that's how they view our research studies. So as far as them being the primary people to return these results when it's really a research goal and not a clinical care goal, insurance is not going to be billed for that. The physician's not going to get paid if insurance isn't billed for that. And so how do they get paid? And so I would say that because I run a clinic and I watch the RVUs, that my physicians would not be enthusiastic at all about returning research results when there's no compensation for that activity at any scale.
Deborah?
So both CSER and eMERGE were set up specifically to look at genome sequencing in a clinical setting. So you were anticipating returning results, but there is also the situation where a participant is requesting their results where that return of results isn't planned as part of the study. How does that change how you think of return of results when it's a kind of pull situation rather than a push with a choice situation?
Maybe not completely on target to the push versus pull, but and Gail was very actively involved in this as well. In eMERGE 1 at our site at Northwestern, one of the things that we encountered was several, I want to call two handfuls worth of participants for whom we knew that they had a likely highly penetrant HFE variant. And we had specifically said we were not going to, in eMERGE 1, we had specifically said we were not going to turn results because of this question of we didn't even know if it was going to work to use electronic health records data. So in those cases, we actually did go back to the IRB and request permission to return results. Turns out about half of the people that we determined had an HFE variant already had a diagnosis of hemochromatosis, so they were fine. And for the others, they were very happy to get that information. Now, hemochromatosis is not, maybe as scary a disease as some, but that worked very well for us. So, and that was one example where even though we had said we were not going to return results, we got permission from the IRB, did it, and both the patients and the treating physicians were happy with that outcome.
Yes, so we had the same experience with hemochromatosis, and I will object to the not as scary a disease. It's a lower penetrance disease, but I've actually seen patients die of hemochromatosis because it was recognized too late. When they had end-stage disease. So I've had this happen in I've been doing research for a long time, and there have been a number of studies that we've actually not anticipated returning results, and that we found results as technology changed that we did believe should be shared with participants. We went back to the IRB in those cases. And what we did is we offered return of results either to entire cohorts, because you can't just send 10 people letters saying, do you want your results, if you're not sending everybody a letter, because then they know there's something there. So either we consented the whole cohort, or in cases of very large cohorts, we basically did at least double the number of people. So we did both positives and negatives and asked them to reconsent for return of results. So we didn't want to tip off that so you don't want to call someone and say, hey, I've got something, do you want it, right? That's a little bit of arm twisting. But we wanted people to really feel free to say no to these results as well. But we've had good cooperation from the IRB, and in general, participants have been very interested in the results.
Chester?
Yes, I have questions about scalability. And so as we're thinking about potentially opening this up and all research information is given to subjects, clearly there's a big genetic professional shortage, right? And you have the luxury of having a research counselor and a clinical counselor, great, but a lot of places obviously don't have clinical counseling. I know that there are a number of proposals regarding computational solutions, interfaces for clinicians, for patients. The genome data, other personal health information goes into this to help educate. So is the big picture, as this grows, what would you see as some of the potential implementation challenges of these sorts of tools?
Well, I guess the first starting point is that as of today, the tools are still probably not where they need. I think it's fair to say the tools are not where they need to be. ClinGen, in conjunction with CSER, actually, I should let Gail talk about this, but did a really interesting study in terms of just how good these annotation tools work, and they didn't work so well. And the results of the ClinGen CSER was to think about developing tools that would improve the quality of the clinical annotation. But, you know, these days I would say, and we've heard this a couple of times in the meeting, clinical annotation is much more complicated than actually doing the sequencing itself. And so I think a variety of tools, and I would point to the work that ClinGen is doing in particular to think about sort of automated learning and taking advantage of artificial intelligence kinds of things to improve the quality. But I think that's still a ways out. We're not there yet.
Even with, I'm saying things we can interpret, so not the clinical validity issues of variants of uncertain significance, but rather simply creating things that will allow, you know, the primary care physician who doesn't know genomics to go in and kind of easily, not easily, but in a reasonable way, interpret things as they come in. Those kinds of approaches and tools. Do you see any pitfalls, cost, anything that we might have to contend with?
So I think my experience is that things that are extremely well known in science and genetics are not uncommonly misinterpreted by primary care providers. And I think we have a very, very long way to go to educate primary care providers about how to implement genomic results. And, you know, I cannot speak to the other kind of research results that this committee might be facing. I think there will be tools to make it easier, but the simplest things are gotten wrong. And there is a huge need for genetics consultation around these things. And, you know, we have people coming into genetics clinic with their 23andMe reports, and they want them explained to them. 23andMe says you have a 40% risk of Alzheimer's disease. They are not happy they ever asked the question, honestly, which is, I think, something we need to think about on the consenting side. How do we consent people to understand what they might get back? You know, people tend to say they want everything, but then when they get it, they're not so happy about it. But medical geneticists are not available to explain every confusing recreational genomics test or every research result. And also, those costs are now transferring to the clinical healthcare system, right? Because you're billing your insurance company to have your 23andMe or your research results explained to you. And so I think there is a caution there about what we decide to share with people, especially things that have no clear meaning for healthcare. We do actually give people their uninterpreted genomic data back when they ask, their BAM files. But you know, those are usually very sophisticated people. We don't offer them routinely. We actually wrote a study to try and explore that, and we didn't get it funded, so maybe you could suggest funding for exploring that. But I am concerned about who's going to explain these results, and the idea that these results interpretations will change over time. Who's going to do that work? And if it's not in the research study, if it's not in the healthcare system, where is it? I don't know.
I would just add a couple of points to that. I think one of the challenges that we need to overcome is conversion of at least genomic results from PDFs into actually computable lab values inserted in the electronic health record. And it's one of the things Emerge is working hard on being able to actually do. Once you do that, then you can be in a world where you actually fire clinical decision support just as you would for many other non-genetic lab values. But we're in this bizarre world where all of genomics is so heavily driven by computers, and then the testing labs give you a PDF to work from. And so I think that's a good area. The second point that I wanted to make is I think to, again, look at things like ClinGen and the idea that there will be an emerging knowledge base that you could then somehow integrate and filter genetic information that is present on your participants against that knowledge base in an automated kind of way, and then fire clinical decision support. I think that's the way forward. And then maybe at that point, primary care physicians might feel more comfortable.
We have about 5 more minutes, and I see 3 cards up, so we'll try to get through all of them. Rhonda?
Yes, Gail, I want to go back to something you said earlier. You said there's not been a legal challenge to whether CLIA is the standard for returning non-CLIA accredited results, but that your institution let you return some results. Was that one-off because your CSER and eMERGE studies are so rigorous, or was there an institutional structure for deciding, and how was that put in place?
Our IRB, we have a number of committees for our IRB, and they make different decisions. And within the same institution, you would get different results. And so I'm happily in a committee that understands genetics, and they are very protective of the participant's best interest. And so if they feel that it's in the participant's best interest to get that result, then they will share that result.
So your IRB had the expertise to judge the reliability and validity and chain of custody of your samples?
We have enormous genomic strengths at the University of Washington, and our IRB believed our capacity to correctly interpret the results. But our IRB is somewhat proactive in this field, and actually Dr. Fabsitz led a paper a long time ago now on return, which was the first paper to really advocate for sharing genetic results with research participants, and my IRB sent it to every single person doing a genetic study at the institution, including me, to make sure we were aware of it, and I was a co-author, so I let them know I was aware. So because we have an enormous amount of genomic research at UW, we may be in a unique position.
Richard?
We've talked almost all morning about returning clinically actionable results, but in some of the readings that we had for today's meeting, there are people who said, I only want the good news. Have you had any experience with people asking for just the good news of their genetic results, or how do you react to something like that?
I'm not sure how you define just the good news, because actually we're rarely offering good news. We don't do ancestry testing, which is extremely disappointing to all of our participants. We offer them highly penetrant disease-related pharmacogenetics and carrier testing in general, and we offer them in the categories. Most people take all of them, and I've seen that at other CSER sites as well, that most people take all of them. We don't do associations with you're taller or you're, more athletic or whatever. That's simply not something that we offer.
Yeah, I think it's hard to know what good news really is. I mean, is good news the absence of a highly penetrant BRCA variant? That still doesn't protect you against breast cancer. So, one of the things in the data, it's too early to analyze the data, but in our eMERGE site at Northwestern, we've got sort of 2 arms to our study. One arm is returning all 109 genes that are on the panel. The other arm is an arm where we've actually told people, we're going to give you the ACMG 59, and then you can tell us, do you want cancer gene susceptibility genes? Do you want, metabolic susceptibility genes, et cetera? And so the data will come, but at least we'll have some data about the types of genes that people might want to get information back about.
Greg, question and response in the last 2 minutes.
Yeah, so it's interesting listening to this because genetic medicine is not disease specific. And coming from the HIV field and also listening to Fran this morning, breast cancer, a lot of these issues are the idea that you're going to give people or you're going to decide for them what's important. It seems much more top-down than bottom-up in the way HIV and breast cancer care is developed, and where some of the interpretation of results is borne by the communities themselves. Working in Africa, we train people in rural areas about virology and immunology, and maybe not as sophisticated as people in this room, but be able to make clinical judgments with their care providers based on this knowledge. And so I think there's going to be some distinction between when we're talking about research results that are in a non-disease specific context, where there aren't sort of systems of care that are developed, some some of these tools to deal with research literacy, treatment literacy, and the more general kinds of medical information that genetic studies are going to provide.
Well, thanks for a terrific panel, and thanks to the committee for your questions and comments. We now have a panel to be led by Dr. Chester Brown, who's professor in St. Jude Chair of Excellence in Genetics, Department of Pediatrics at University of Tennessee. So I'll turn it over to Dr.